Hello everyone and welcome to today's webinar, Balanced HEK293 Viral Feed, Accelerating AAV and LV Production for Gene and Cell Therapy. I am Cassie Soltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by FUJIFILM Irvine Scientific. To learn more, visit www.irvinesci.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You can also use this if you're experiencing any technical issues as well, and it's located directly beneath the presentation. I'd like to now welcome our speaker, Shan Gao, PhD, Senior Scientist 2, R&D Bioproduction at FUJIFILM Life Sciences in Irvine, California. Dr. Gao, you may now begin your presentation.
Thank you everyone for joining us today to learn about balanced HEK293 viral feed in accelerating AAV and LV production for gene and cell therapy. For those of you who are not familiar with FUJIFILM Irvine Scientific, we're part of FUJIFILM Life Science Division, specialized in cell culture media development, aiming to empower all who bring medicines and treatments to life with unmatched quality and responsiveness. We're headquartered in Orange County, California, where our R&D and original manufacturing sites are located. In the last few years, we have expanded our manufacturing and operations to East Coast US, Europe, and Asia to meet the market needs and offer our products and services to the global customers more efficiently. As to the industry we're serving for, viral vector production, there are three main categories depending on the clinical applications, including cell therapy with lentivirus as the most used vector, gene therapy with AAV and herpes simplex virus among a few FDA-approved gene therapy products, and last but not the least, live or attenuated viruses used in vaccine production. The common goals for the viral vector manufacturers and their suppliers are to reduce production cost, improve yield, and improve viral vector quality. The study I'll be presenting today will focus on gene and cell therapy applications, specifically for AAV and LV or lentivirus production. I like to use this transient transfection process as an example to further inspect the AAV manufacturing workflow because it is the most used platform so far in viral vector production. Different strategies have been applied or developed to optimize each step of the process, including cell line development, plasmid vector design, transfection reagent, growth media, downstream purification, formulation, etc. In the meantime, we also understand that once a process is developed and locked, it is risky and costly to introduce any major modifications, including aforementioned strategies. Therefore, our solution is to introduce a feed media that could be compatible with various viral vector production platforms in a fed batch format. With a simple addition of balanced HEK293 viral feed 24 hours post-transfection with 12% of the culture volume, it will improve viral vector production yield with minimal cost. To this goal, we designed a series of experiments to study the compatibility of balanced HEK293 viral feed with various AAV and LV production systems, including different cell lines, growth media, 3 types of transfection reagents, culture formats, and different AAV serotypes. This is an illustration of the AAV production workflow that we used in the shake flask in a fed batch format. Following cell thawing and adaptation to specific growth media, cells were seeded at 1 million cells per ml and triple transfected about 24 hours later with AAV plasmid and the transfection reagent. 24 hours post-transfection, viral feed was applied with 12% of the culture volume. The cell culture was harvested 72 hours post-transfection. The harvested material was used for titer analysis. The analytical methods we used for viral capsid titer in cell lysis include traditional ELISA, biolayer interferometry-based immunoassay, and automated immunoassay. For viral genome titer, we used qPCR in our early development and now completely switched to nanoplate-based digital PCR. The viral genome titer results from our collaborator was collected from digital droplet PCR assay. This was the first set of data from the early phase of the development, showing the performance of the balanced HEK293 viral feed in comparison with other feeds in our feed panel. The growth media for all the cultures were balanced HEK293. We had no feed control and other feed formulations. From the gray bars, we can see balanced HEK293 viral feed improved viral genome titer about 10 times over no feed control. The improvement was significantly greater than other feed formulations. This set of data shows the improvement of AAV2 viral genome titer in 3 cell lines using the viral feed over no feed in shake flasks. The gray bars indicate the titer without any feed for each cell line, and the green bars indicate the titer with the viral feed in each cell line. The first 2 cell lines are the most used viral vector production in the industry. The third cell line is our in-house adapted suspension 293 T cells from adherent 293 T cells. Cell cultures were in triplicates for each culture condition, and PCR was performed in triplicates for each sample obtained from each cell culture. The data for the first 2 cell lines are from qPCR assay, and the third one is from digital PCR assay. As you can see, the viral feed significantly improves AAV2 titer in all 3 cell lines at different degrees, specifically by 3 to 8-fold. We also would like to verify the effect of viral feed in bioreactors. First, we tested in microbioreactors. In this experiment, we like to optimize the propeller speed for transfection as well as the performance of viral feed. Again, the basal media is balanced HEK293. Gray bar stands for AAV2 viral genome titer for no feed cell cultures, and green bar stands for the titer for viral feed cultures. We also had a side-by-side control of cell cultures for AAV2 production in shake flasks. All cell cultures were performed in triplicates. In both propeller speed, viral feed significantly boosted AAV2 titer by 3.5 to 4.5-fold over no feed cultures, which is consistent with the effect in shake flasks. Next, our collaborators tested the viral feed performance in 2-liter and 10-liter XDR bioreactors using their platform, a different HEK293 derived cell line and AAV plasmid system using balanced HEK293 as a growth media for AAV2 and AAV5 production. The viral genome titer was measured by digital droplet PCR. The plot on the left shows 2-fold improvement for AAV2 production with a viral feed in 2-liter bioreactors over the culture using balanced HEK293, the growth media as a feed. As to the data in the plot on the right from 10-liter bioreactor experiment, because of the limited bioreactors they could spare for the experiment, the culture with viral feed was performed in 1 bioreactor, and 2 cultures in the shake flask with or without viral feed were performed side by side as controls. The improvement of AAV5 production with a viral feed in the 10-liter bioreactor is about 1.5-fold over the no-feed culture in a shake flask. Our collaborators also kindly helped us test the viral feed performance for different serotype AAV production in shake flasks. In this experiment, the improvement with viral feed is different depending on the serotype: 4-fold for AAV2, 3-fold for AAV5, and 6-fold for AAV9 comparing to the non-feed control. Although PI-based transfection is most used in viral vector production by transient transfection, we'd also like to test the viral feed performance for lipid-based transfection reagent. We noticed the commercially available viral vector production system that not only uses a lipid-based transfection reagent, but also consists of optimized cell line, growth media, and lysis buffer. We really don't know what to expect before applying the viral feed to this optimized system. This set of data shows twofold improvement of AAV2 viral genome titer with balanced HEK293 viral feed over no-feed culture using all the components from the optimized system. Although the improvement is only double of the no-feed culture, we're pretty happy with the result because it demonstrates the potential of the viral feed to improve viral vector production even for an optimized system. When we consider the compatibility of balanced HEK293 viral feed with different viral vector production platforms, the growth media is a major factor. We understand the efforts have been taken by our customers to choose the growth media that works best for them. Therefore, we tested 4 other commercially available media for suspension HEK293 cells besides our balanced HEK293. Suspension HEK293 cells were adapted to each media for AAV2 production using PEI-based transient transfection. This result demonstrated the improvement of AAV2 titer with balanced HEK293 viral feed over the no-feed culture across the board of all the basal media tested. The improvement is between 2 to 8-fold depending on the specific media. We also received inquiries from some customers about the possibility of using the viral feed on adherent cell cultures because they prefer adherent cells for viral vector production for a number of reasons. Therefore, we tested adherent 293 T cells for AAV2 production using the viral feed. The plot on the left shows 3-fold improvement of AAV2 viral genome titer with viral feed over the cultures without any feed. In addition, we also measured the capsid titer using BLI-based assay in the plot on the right, showing in the green bars. We noticed less improvement of AAV2 capsid titer than the viral genome titer improvement for viral feed cultures over no-feed cultures, which indicates potentially higher ratio of viral genome titer to capsid titer. If I could read your minds, you were probably wondering how the capsid titer would change with the addition of viral feed for suspension HEK293 cells. Here's a summary of AAV2 viral genome titer and capsid titer for suspension HEK293 cells cultured in 3 different culture vessels, including 125 ml shake flasks, 24 deep well plates, and 96 deep well plates. The data showed more increased capsid titer than viral genome titer, resulting in reduced percent full capsid. The titer data was collected 72 hours post-transfection as the standard protocol, but we were wondering whether the addition of viral feed would change the AAV production kinetics throughout the production process after transfection. Therefore, we set up a study in benchtop bioreactors to map out the AAV2 production kinetics after transfection, side by side with a bioreactor using viral feed and the other one without any feed. We also set up the similar cultures in 125 ml shake flasks. The shake flask runs were in duplicates and bioreactor runs were 1 for each condition. As we noticed, the overall higher AAV production in bioreactors over shake flasks in both conditions. We also noticed VG titer reached plateau about 48 hours post-transfection with minimal increase afterwards, or even decrease for the viral feed cultures in the bioreactors. However, the capsid titer showed quite different trend with continuous increase up to 72 hours post-transfection, especially for viral feed cultures in both bioreactors and shake flasks, as indicated by the solid lines. Therefore, the addition of viral feed changed the AAV2 production kinetics for both viral genome titer and capsid titer in this case. To achieve the optimal VG over CP, or percent full, 48 hours post-transfection is optimal harvest time rather than the 72 hours. As we have been using PEI as our standard transfection reagent, so the cell growth after transfection showed significant difference between the 2 transfection reagents with VR transfected cells barely grew after transfection as to the viability at harvest. VR transfected cells showed the lowest viability, around 75%, compared to PI transfected cells with over 85%. However, for the cell cultures transfected with VR and received the viral feed, the viability at harvest increased significantly to about 90%. As to the viral genome titer and capsid titer, we saw as to the VG over CP, there's quite an interesting result. PI and VR transfected cells showed similar VG over CP. The addition of viral feed reduced VG over CP of AAV2 produced by cells transfected with PI, but increased VG over CP produced by cells transfected with VR. Therefore, the combination of transfection reagent Verisgene and enhancer Revit would result better overall AAV2 production in terms of VG titer and VG over CP in this case. Next, I'd like to switch gear and talk about the application of balanced HEK293 viral feed in lentiviral vector production. And the process is very similar to AAV production, except we're using 4 plasmid systems for lentiviral vector generation instead of 3 plasmid for AAV. And again, viral feed is also added 24 hours after transfection with 12% of the culture volume. And for harvesting, we use centrifugation to separate cell pellet and the supernatant, and we'll collect the supernatant for titer measurement. As to the lentiviral RNA titer measurement, after extracted the lentiviral RNA, we use the DNA digestion to remove any background plasmids before setting up the digital RT-PCR. The lentiviral RNA titer showed threefold improvement from cell cultures with balanced HEK293 viral feed compared to the cultures without any feed. We're happy with the result, but we'd also like to verify the functional titer of the lentiviral vector produced under both culture conditions. Therefore, we measure the infectivity titer based on the expression of GFP in adherent 293 T cells. Specifically, we seed the cells in 24 wild plates on day 1. The next day, we transduce the cells with serial dilutions of lentiviral vector samples. We also counted the cells using 3 vials of the cell culture for infectivity titer calculation later on. I just would like to note that we noticed some matrix effect from the harvested and non-purified samples on viral vector transduction efficiency. So we use the LentiX concentrator to replace the spent media from the harvested lentiviral samples with PBS. Although we didn't use it for the purpose of concentrating the viral particles, it provided us a quick and easy method for buffer exchange to eliminate the matrix effect on this functional assay. 72 hours later, we harvest the cells for flow cytometry analysis to quantify the population of GFP positive cells from each well of cell culture. The infectivity titer was calculated based on GFP positive cell population and cell number at the time of the viral vector transduction. The lentiviral infectivity titer also showed over threefold improvement from cell cultures fed with balanced HEK293 viral feed compared to the cultures without any feed, which is consistent with the viral RNA titer, indicating comparable infectivity but higher yield of lentiviral vectors using balanced HEK293 viral feed. With both viral RNA titer and infectivity titer, we are confident that this viral feed indeed
Okay, wonderful. Thank you, Dr. Gao, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located below the presentation, and we will answer as many of your questions as we have time for. Our first question asks, it says, you mentioned applying the viral feed to cell cultures 24 hours post-transfection with 12% of the culture volume. Have you tested other concentrations and time points of adding the viral feed to cell cultures?
Thank you, Kathy. Thank you for the question. We actually get this question quite often, so I explain over here. So first about the timing. So we know this viral feed is not compatible with transfection, so that's why we recommend 24 hours after transfection for just the easy implementation. But some of our customers tried 12 hours and 24 hours, so it's really depending on whether you would like to explore other timing in terms of your specific system. But we would recommend at least 4 to 6 hours after transfection, so it won't interfere with the process. And in terms of the concentration, because this is such a rich media, so the concentration is kind of limited by not increasing the culture osmolarity after you add it in. So we didn't test the other concentration because this is really what we designed for, but some of the other customer tested 6% to 12% and found out 10 to 12% works the best. As I mentioned earlier, if you decided to do like a bolus, 2 bolus feed at 12 hours and 24 hours, then you can try 6% for each. So yeah, hope it answers the question.
Wonderful, thank you. Okay, our next question asks, does the viral feed work for vaccine production through a transduction or infection process?
Yes, thank you for the question. So while we don't have the capability of like a model system for vaccine production for infection process to produce viral vectors, but some of our customers do use the viral feed in their process. And I think it's really depending on the specific system or what kind of viral vector you're producing. So if you're interested in it, feel free to contact us, and we may be able to offer some samples for a small-scale study to see if the viral feed would help with your system.
Okay, wonderful. Thank you. And again, if you have questions for our speaker, go ahead and put them into the Ask a Question box located just below the presentation. We'll move forward here with our next question. This question asks, can this product be used to achieve high cell density cultures?
Oh, thank you for the question. So even though this feed is specifically designed to improve the viral vector titer instead of promoting the cell growth, but we do have options. If you're interested in like intensive process, especially for HEK cells, please contact us afterwards. We do have some other options we can discuss. Thank you.
Wonderful, thank you. All right, and I'm not sure, we are working on getting a couple of slides here uploaded. I know there was something you wanted to go over for the audience. Did you want to speak on anything regarding your presentation as kind of a follow-up statement?
Yes, so I think there's some issues with a few slides, especially about when we're trying different transfection reagent. So we really explore different systems over here. Even though our model system is PI and PI Pro, which really based on the industrial standards, but we do want to cover some newly on the market transfection reagent just to see whether it will be compatible with our viral feed. Yeah, so it's that part of the slides I would like to go over. Yeah, I can talk even without the slides,
Okay.
if possible.
Yeah, while they're uploading,
Yeah, so
getting them loaded. So if you just want to discuss, and then once they're up, we'll show them for the audience.
okay, yeah, so instead of doing it in the shake flask, we carried out that specific experiment in the 24-well plate. As you can see from our previous experiment, we compare with the shake flask, 24-well plate, and 96-well plate, and it really showed comparable result across different cell culture vessels. And 24-well plate is really our go-to experiment system because it's compact, they use less material. Are we on the slides now?
Yes, we are on the slides.
Yes, I think. Okay, yeah, this is slides I was talking about. So it really allowed us to do some more different conditions and to test everything side by side instead of handling a whole bunch of shake flask over here. So that's why we picked the 24-well plate. And in this specific experiment, we used the PI and as well as VirusGene and Revit. So VirusGene is a transfection reagent and Revit is an enhancer. And just for simplify the name, I just call it VR. So we used PI and VR transfected cells, either non-feed or feed in this system. And if we can go to the next slides, and that will be the cell growth data. Okay, so from here you can see there's really different, quite different cell growth pattern, especially after transfection. So the VR transfected cells, either feed or non-feed, they grow, barely grow. But for PI, you still see quite a bit grow after transfection. However, there's some also interesting result from the viability. So for the cells just transfected by the VR without any feed, you see like really low, pretty low viability at the harvest 3 days after transfection, around like 75%. However, for the cells transfected by the VR but received a viral feed, you would see there's a significant improvement of the viability at the harvest, almost comparable to the PEI transfected cells. So we thought that was interesting. And if we can go to the next slide. Okay, so this is the result for the VG and CP titer. As you can see, I know the scale is very skewed because significantly higher titer improvement for the VR transfected cells. So you barely see any improvement, but yeah, there's some improvement for the PI transfected cells for the viral feed compared with no feed. Yeah, so the good news is for both of the transfection reagent, viral feed was able to improve the VG titer. Yeah, even though the VR is way higher than the PI. And so that's for the VG titer. And when you look at the CG titer, it's pretty much similar story over here. I think next slides will be, I think that we're fine with the VG over CP slides. So yeah, so this is what I would like to clarify.
Thank you. I think that's really great to have that up on the screen for our audience here. And then I did want to bump back over to Q&A. We did get another question submitted before we end up wrapping up. This last question here says, we're using Thermo Fisher production kit for AAV2. Can I use this for this production system?
Yes. The short answer is yes. Yeah, that's why we tested some of the optimized, like you can call it like kiddish AV production system, because we want to verify like what's the potential of this viral feed. Can it be used for already optimized AV production system? And we do have some success in it. But I think it also depending on your vector, the different serotypes or different kind of like transgene you're trying to express. So yeah, if you're interested, please reach us, and yeah, we can offer some samples for a small scale study.
Wonderful, thank you. I want to thank you for your time today. We would also like to thank LabRoots and our sponsor, FUJIFILM Irvine Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for and those submitted during the on-demand period will be addressed via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.